All right, so welcome back to session 2 of our Intro to AI for Science on supercomputers. Today, we're going to have Dr. Bethany Lush talking to us about neural networks on Python. We're also going to be having a science speaker at 4 p.m. Central, Austin Clyde, come tell us about the work he's been doing using AI in therapeutics. And with that, I'll have Bethany start us off.
Thank you. So I'm going to share my screen and bring up our GitHub repo. But I want to start with asking if people had any questions about forking the repo. We talked about how you can clone it, but to do homework in the repo, you could fork it and submit it. I know last week that you had the option to do Google Colab instead.
And as we did last week, as you have questions, obviously, please put them in the Slack workspace. It's easiest for us, and sort of go from there.
Okay, so I'm going, let's see. I'm going to show you my browser. Okay, so I navigated to our GitHub repo. Sorry, did someone say something?
I think you're good to go, Bethany.
Okay. Yes, we still, theta is unexpectedly down longer than we thought today. So we're going to use Google Colab again. But if you've already forked the repo, then you might want to pull our updates. So last week, Taylor had instructions for syncing your fork, and I linked to them in week 2. And so I'm scrolling down here. So you can do this git remote add upstream and so forth. But for the purposes of Google Colab, we can just, let's see. If you've cloned your GitHub repo somewhere, you can update it right now with git pull or with syncing your fork. If you just want to directly open our notebooks in Google Colab, we can do it the same way as last week. So we're gonna start with this first notebook, but have a little bit of introductory material. Last week, Taylor introduced you to machine learning, and this week we're going to talk about neural networks, which is a technique within machine learning. And you'll hear people refer to this back and forth with deep learning. Roughly, deep learning refers to when you have a neural network with enough layers, but we'll just use these terms interchangeably. And this week, we're going to build up the understanding of neural networks by going through the math and writing it out by hand, in the sense that we're just going to use like NumPy. And next week, you'll learn how to do deep learning with these nicer Python packages like TensorFlow. And so it's you have higher level functionality, and a lot of these details you don't have to implement yourself. So we can go to Google Colab, and we can do this GitHub tab, and I can click on the first notebook and paste it into search. Okay. And if you want to be fancy, you can even say you want a GP. Okay. If anyone's having questions with, you can see I'm at collab.research.google.com, and you can see I've pasted in the GitHub repo the link to this notebook. If anyone has questions about that, you can post in Slack. Things seem okay so far?
Yeah. A quick question. How do you get to that?
To this page?
Yes.
The particular notebook. Okay, so we're using this GitHub repo for the whole series. We did 0, 0, and 0, 1 last week. Now we're on 0, 2, neural networks in Python. And then I clicked on the first notebook, 0, 1. And I copied the URL over to Google Colab. So there was that GitHub tab, and I could paste in the URL and press search, and then it brought up the notebook in Google Colab. Okay, so we're going to talk about a basic kind of neural network that's sometimes called a multi-layer perceptron model. And the machine learning goal this week is to classify handwritten digits. This is a supervised learning problem, which Taylor, I think, touched on that terminology last week. Supervised learning means that we have inputs paired with outputs. So you're learning, I guess the supervised part is that you've got these labels. So in this case, the input would be a handwritten digit, and the output would be its label, which digit it is from 0 to 9. And we can represent this image as a matrix with entries for each pixel. They're grayscale, so the entries are from 0 to 1. So I have like a little image showing how the image goes to a matrix. And in our machine learning goal is to find some function that will map this image to the label. And this would be helpful, let's say if you're at the post office and you want to automatically sort our mail by zip code, you want to be able to read people's handwriting of the zip codes. And this is a real early success story of machine learning. So first we're going to import some Python packages. Now, I included TensorFlow just for one convenience function. Yes, a question.
Yes, hi. I can't find this notebook.
Like on this page, you don't see it?
Please.
Argonne LCF.
For this kind of question, please address the Slack. We won't be able to stop and help everyone who needs help accessing. If you are not part of the first session and haven't reviewed first session, you may have some troubles getting started again. So just
I found it. Got it. Thank you.
Perfect.
Okay, so I'm the TensorFlow package we're going to import because it conveniently has this data set handy. And here we can see what it looks like. So you can see there's some pretty bad handwriting in there. They've got some pretty crazy 9s and that sort of thing. And here's a convenient function to load the data. And so the first time you run this cell, it might take a bit. It actually was really fast for me. Now, by convention, we're going to call the inputs X and the outputs Y. And again, the inputs are the images and the outputs are the labels 0 through 9. And this convenient function already has the data split between training and test. And training data is what we can use to fit our function. And the test data we hold out until the very end. And we use it to estimate how well our model will generalize to new images. And here's some common pre-processing on the images. We're going to convert the images to float because currently they're integer pixels. We're going to divide them all by 255 because currently they're this grayscale from 0 to 255, and the neural networks work better when we rescale to something like 0 to 1 or -1 to 1 or something like that. And we print in this shape. So there's 60,000 training images. Each of them are 28 pixels by 28 pixels. And for today, since we're going to do a simple neural network, we're actually going to flatten each image into a vector. So it's going to be 28 times 28 long. But next week, we'll talk about how to leave the images in 2D with a different kind of neural network. So now that we've flattened them, our training data is 60,000 images, and they're each a vector of length 784. And we'll do some simple processing on the labels too. We just want them to be integers. And so we can check again that the training data is, there's 60,000 training examples, there's 10,000 test examples. The training data, each of them is a vector of length 784. And the labels it's just one vector. There's a number, it's a number for each image, and it has the digits 0 through 9. And this cell will plot the first 10 of them and label them with their class label. So you get some idea that there's some variation in how people write the same number, and we can see that you can check this vector Y-train to see what the class label is. Now, last week, Taylor talked about linear regression. And in that case, the input was just a scalar, like just a 1D input, 1D output. This time, we're going to start with linear regression again and move up to nonlinear. But this time, the linear regression is on a higher dimensional input because each input is a whole image. And that was a vector of size 784. So instead of multiplying by just a slope, we're multiplying by a whole matrix, W. So here is like the simplest thing we could do, a linear model. We're just have the images and we multiply by a matrix. And like last week, we can start with a loss that's mean squared error. So here, to evaluate our model, we can plug in the data and then do mean squared error. So that's just like last week. And like last week, we can start with a gradient descent. So here's a variant, another rewriting of the same thing like you saw last week. We can check the number of examples that we need to average over. We can apply the current model to the data. Here's the gradient for this mean squared error for linear regression. And here we're updating the weights by taking one step forward. And like you saw in your homework, you don't necessarily want to use all of the training data in each step. So for the moment, I'm just going to pick the first 100. And I'm going to loop along and keep improving the weight W iteratively, and we can plot the loss over time. So we can see it improves drastically at the beginning and then starts to plateau. Looks like if we ran it longer, it might keep improving. Okay, but we can check how it's doing so far. So here I'm applying this linear model to the first 10 digits and see what the labels are. So as you might expect, it's not doing very well. So one thing is, you notice it's predicting real numbers. So that's weird. We want to predict classes, 0 through 9. And also, even if you round them, most of them are wrong. So that's not too surprising. We just have a linear model. Okay, so I think let's pause now. The mean squared error might not be very intuitive for a classification. And it's often more intuitive to look at accuracy, which is the percentage of images correctly labeled. So let's pause, and you can implement in the next cell accuracy. So number of images correctly labeled, and then we can talk about how we could also plot accuracy while the training is going on. And this can be also an opportunity to catch up if you're having trouble loading the notebook. Okay. So this would be one way to define accuracy. I'm trying to match the format of how we did the loss up here. So I'm passing in the current parameters, the input data, and the true labels. So here I passed in the parameters, the input data. I'm predicting these labels. Now we should do some rounding. So the true values should be a vector of integers, and this should be a vector of integers. This will check how many of them are equal and sum that up, which is one way to count how many of them are equal. And then I'm dividing by the number of images to get a percentage that are correct. Okay, as you can see, I was adapting that on the fly, so let's hope that works. So we could plug this into our training loop. And it can be another thing that we can plot during training and see something more intuitive. Any questions about this? Okay, so the loss is like smoothly going down, but in this case, the accuracy is a lot less smooth. And in my opinion, more intuitive. So we would get the idea that near the end, something like 35% of the images are accurately labeled. Any questions, or did anyone catch any mistakes? Okay.
I had a quick question. Should we also include something in the model to make sure that it doesn't predict, for example, a negative value for the 0 class? Because right here it predicts -1 as a score.
Yes, that is a great point. So I'm going to talk about that next, that this model doesn't make a lot of sense for classification. And so I'm going to introduce some improvements to the model that make more sense for this problem and make the model more complicated. So one thing that we could do is we can add a bias term to make it more complicated. So in the 1D case like last week, that's like the slope intercept. In the higher dimensional case, this is adding a whole vector. So that's typical. And then, yes, this excellent point that we really want it to output integers, not real numbers. So this is like going from linear regression to logistic regression, if you're familiar with that. And something other than mean squared error might give us better results. So one thing we can introduce is the sigmoid function, which you might have seen before. This formula might not be very intuitive, but we can plot it. And we can see that it's a function that goes between 0 and 1. And so if we tack that on to the end of our model, then at least we're predicting things from 0 to 1. Now, wait, one more step to, because you're going to say, wait, I want things 0 to 9. One more thing. Okay, so for the moment, let's just look at this line. Okay, so this part's the same as before. We multiply by a matrix. Now we're adding on that extra piece that we're going to add a vector, which people call a bias vector. And then we're applying this sigmoid, which means our outputs are going to go from 0 to 1. But really we want to predict classes 0 through 9. And so one common way to approach this is called a one-hot encoding. So instead of our labels being a vector that's just digits 0 through 9, for each image, this is easier to show you than to explain. For each image, we have a vector label. It's length 10 because we have 10 classes. And the index for the correct class is a 1, and the rest of them are 0. So one hot, one of them's on, the rest of them are off. And so here we're using this convenient function to just do that. We can go from the vector of labels where we had 0 through 9 for each image. We tell it how many classes we have, which was 10 because we have 10 digits. And this will conveniently transform our labels from just being a vector to being a matrix with 10 columns. And here I'm printing the first 10 examples as a demonstration. So now we could imagine that instead we want our model to output a vector of length 10, and we apply that sigmoid. So now you're predicting 10 numbers, and they're all bound from 0 to 1. So now we're getting closer to something that makes sense for classification. A further improvement is it's common, instead of using sigmoid at the end, to use softmax, which is similar but makes them all add up to 1. So it's got this this normalization here. And so now we're outputting for each image a vector of length 10, where each entry is between 0 and 1, and the entries add up to add up to 1. So you've got something like a probability distribution for each class, and the entry with the highest value is the class that you're predicting. And to demonstrate how this looks in math, here I've updated our model to be that we multiply by W, add the bias, and then apply softmax. And I'm going to start with just a random initialization of W and B. And we can apply that model to some of the examples and print the output, just so you can see what I'm talking about. Now, in this case, we didn't actually train this model, we just defined it and we started it with random weights. And so here, it's predicting all of the classes to be about equal because it really doesn't know anything yet. We haven't trained this model to know things about images. But you can see the idea and that they sum up to about 1. So these are iterative improvements on the neural network to make more sense for classification. Now, another improvement in our previous training, the thing that we were minimizing was mean squared error. And so that's what the gradients were all based off of. For classification, it's common to use something else like cross entropy, binary cross entropy for 2 classes, or it's called categorical cross entropy if you have more classes. So I'm not going to go into like the reason it makes sense, but it connects to information theory. You can read about it more here at this link. But just wanted to make you aware that that is a common loss function when you're doing a classification problem. Now, you could say, why don't we use accuracy directly? Because that's intuitive. But if we look back at how we defined accuracy, we've got this a binary, like it's correct or it's not correct. And this is not something that is like smooth that we can take gradients of. Okay. So those were various improvements, but still on the inside, we had just a linear model. Just multiply by W and add a bias. Multiply by matrix, add a vector. And really in neural networks, the magic comes when we need some nonlinearity. So if you think of fitting just a line to a complicated polynomial, or in higher dimensions, fitting a plane to something complicated, we don't expect it to work well for some data that's inherently nonlinear. Now, a basic neural network would be exactly the form we've been talking about. You multiply by a matrix and add a vector. That's called an affine transformation. And then we apply some nonlinear function. People often use sigma there because people, at least used to, often use sigmoid for that activation function. It's some nonlinear function, and for now we'll talk about it being sigmoid. And really we want to stack layers. So we're recursively doing the same thing again. So here we start with that same base, but now we're going to multiply by another weight matrix and add another vector, and then apply nonlinearity again. And then people often draw these layers like this diagram. So the first column would be your input. Then you've got that these connections would be like multiplying by that matrix W. Then you've got the output of applying that layer. And then these connections would be like multiplying by that second matrix. And so here's a diagram showing the math that is in the line above. So if you think of multiplying by a matrix, you might be wondering why are there just like a bunch of lines. With this exact diagram, it could be like your input is 3-dimensional. You multiply it by a matrix that's, since we're doing right side multiply, like a multiply by a matrix that's 3 by 5, and that gets us to this 5-dimensional middle layer that's blue. And then the second layer would be like multiplying by 5 by 2 matrix to get output that's two-dimensional. And so you can think of it as going from this purple representation to this blue representation is fully connected in that each blue circle is some linear combination of all the purple ones, where those weights are the entries in the matrix. And then each of the yellow circles is a linear combination of the blue ones, because we multiplied by matrix. Early, we add a vector, which people don't usually draw in, and then we add the non-linearity. But this is like a convenient way to draw out the layered layeredness of a neural network. So we can keep doing this. Here I wrote the math in LaTeX, but here we can write it out in Python. And now I've got the two layers. We do the dot product of multiplying. Sorry, the NumPy dot can include the matrix multiplication. We do the matrix multiplication with W, we add the vector, apply sigmoid. Multiply by another W, add another vector, apply sigmoid. And we can keep going. We could add a third layer. So that is recursively the same thing again. And in a drawing would be like having another column in our network, which people call a layer. And here's what it looks like with Python. So here we're talking about sigmoid being the nonlinearity, but next week we'll talk about other activation functions besides sigmoid. And you might say this looks like it's going to be a really big mess if you wanted to have a deep network and have a lot of layers. So the good thing is, well, you could think about how to make this in a loop or something, but also the good thing is next week we learn about TensorFlow, where it's much easier to add these layers on. You don't have to explicitly say we're going to multiply, we're going to add, and so forth. Okay, any questions about the form of a basic neural network before I talk about some of the implications? Okay. So to optimize this more complicated model than just linear regression, we still want to do some stochastic gradient descent or a variance of that, but now the gradients are more complicated. So last week it was easier to see what the gradients were to do linear regression with mean squared error. Now we've added these sigmoids, we've added more layers, we might change the loss function to something like categorical cross entropy. So the gradients are more complicated, but it's just a bunch of like linear algebra, basic math strung together that we could do calculus on. And it's a lot of chain rule. And one of the keys to deep learning being successful is making those gradients reasonably fast to compute. And the way this is done is that you move backwards. So we want to know the gradient of the loss function with respect to all these weights and biases, all these tunable parameters. And we start with the output layer and we move backwards through the layers, keeping around some intermediate results to reuse them. And this is called back propagation. And we're not going to go into detail, but that's the idea. And one great thing is we don't have to go through and implement all these details because we use something like TensorFlow. Another cool thing is in TensorFlow, you can start to build more and more complicated networks. And as long as you're using these pieces, these math functions that TensorFlow recognizes, then it knows how to do the gradients, it does automatic differentiation. And so you can like really experiment with building something strange and then count on TensorFlow to know how to do all that chain rule for you. And some terminology is that people often, if you're applying the current network to the data, then it's like you're feeding the data into the input and moving it through these layers. So that's called a forward pass. And then people refer to calculating the gradients as the backward pass. Okay, so adding the non-linearity to your neural network means that you can fit more complicated data. Like maybe your data is like a circle inside of a circle and you want to separate it. But on the other hand, now you're making the optimization more complicated. So in linear regression, when you're doing mean squared error, that objective function is at least convex. If you don't know that term convex, it's okay. It has one global optimum. Now, as we talked about last week, when we get to something more complicated, you can have these like local optima, you can have local minima and saddle points. And so things become important, like picking a good learning rate so that you have a chance to jump into a new area, but you don't jump too much. And there are some variants of stochastic gradient descent that are more effective, like Atom, RMS prop, and so forth. But we're not going to get into that today. They're easier to use in TensorFlow, so you can try them next week. In the meantime, if you're curious, there's some cool animations of how these optimizers do better than gradient descent. Now, another thing to think about with neural networks is now that you've made them nonlinear, you can fit complicated data, but you could overdo it. And you might have seen something similar when talking about fitting data with polynomials in some class before. So here in this picture, we have some data that's the blue dots. And if you tried to fit it with a degree 1 polynomial, which is a line, and it's not going to fit very well, right? Because the data is really curved. So we could call that underfitting. Some people call it a high bias because you're being like very restrictive in your assumptions. Now, if you fit this data with a degree 15 polynomial, it'll do these like crazy curves so that it can hit a lot of those points. And we can see that there's too much variance in there for the real true function, which is orange, which is much simpler. And the balanced thing would be a degree 4 polynomial, which doesn't hit all the points, but pretty nicely matches the real curve. And so the terms I like to use are, if your model is too simple for the data, it's underfitting. If your model is too complicated for the data, it's overfitting. But some people use these terms that are harder, I think, to remember. High bias versus high variance, or you want the balance to be low bias and low variance. So just like you can have this problem with polynomial regression, you can have this problem with neural networks. If your neural network is too simple, like it's only one layer, or it's linear, or the layer is not very wide, like when people talk about layers being wide, that's like how tall this blue line is. and that's like you multiplied by that matrix and it's the output is not very big. So you can imagine that if you made these blue layers wider, then you have way more connections, which corresponds to way more entries in these weight matrices, and a lot more parameters available to tune.
Can I ask a question?
Yep.
So just as a sanity check, so could you scroll to that picture from the first-time programmer block with the like more complex error surface? Yeah, that one.
Mm-hmm.
So when they're the people that are making these blocks and making these pictures, make these animations, clearly they've made the entire, like a large part of this error surface where they show the like peaks and valleys of the error. So in a sense, they've basically mapped out all these different errors, and you can clearly see what the global minimum is from this graph, right? So you wouldn't even really need to do the linear regression. You can just point at the lowest point value and say, oh, that's the one I want.
Mm-hmm.
So the only reason that we can do that here is because they've simplified the problem so that they can actually do this calculation, right? Like normally the reason we don't just do that straight up is because the surface is so complicated that actually calculating it for all of these nodes, I guess you can call them, all these vertices will be too complicated, right?
Yes. So in this case, this weird surface is only in 2 dimensions. So that would be like you only have 2 parameters to fiddle with. But so that could be like when you only had a slope and a y-intercept. But in our case, we could have these weight matrices and bias vectors mean a lot more things to keep changing with gradient descent. And so now we can't plot it. We're in very high dimensional space and we can't explore it all. And so stochastic gradient descent is taking like some path in that space, but we can't try everything.
Okay, thank you.
Okay, so when you fit your neural network to data, you don't just want your neural network to work well on the data you already have. You want it to work well in the future on your future data. So we want to generalize to other data. So that's one reason for holding out some test data, which I'll talk a little bit more about. But also you can think about this avoiding overfitting. And one approach is, or kind of like a general term that covers a lot of things, is regularization. So you're constraining the problem to discourage complex models. Next week, you'll learn about dropout, which is a more sophisticated way to do it. A simpler way is to say that you don't want these weights to be big in these weight matrices W. Oh, sorry, I changed from A's to W's. In these weight matrices W. And so we can add a term to the loss function that is the size of these entries. And that's called L2 regularization. So if we have time, I'll show you the math of that, but that's just like you sum all the squares of all those parameters. Okay.
Hi, Bethany.
Yes.
So I think this is potentially a good pausing point, and then we can sort of jump into a break, come back and hear from Austin, and then jump back into all of this as well. Does that sound okay?
Yes, perfect.
Okay, cool. Okay, so we are going to take a 10-minute break. We'll come back at the top of the hour, 4 p.m. Central, and we'll come back here from Austin Clyde, who's a computer scientist here at Argonne, and then Bethany will continue to share all of her good knowledge about neural networks from there. For those who are thinking about hopping off Zoom, you can just sort of stay on and we'll just put everything silent and go from there. Okay, see you in 10. All right, welcome back. So now we're going to switch gears briefly and hear a science talk by Austin Clyde. So Austin Clyde also works at Argonne National Labs as an assistant computer scientist. And he's going to talk to us about his work using AI in therapeutics. So I'll just hand it off to Austin. If you could, would you introduce yourself, give us a little bit of your background, and then sort of go from there.
All right. Well, hi, everyone. Good afternoon. Let me pull up my slides real quick. All right. Can someone say, yes, they see some slides?
Looks good.
Okay, great. So hi, everyone. As mentioned, I'm an assistant computational scientist here at Argonne. I work primarily on AI problems focused on science, so leveraging big machines to solve big problems. So I'm going to talk to you today about my work in AI for drug discovery. So a bit of background about drug discovery. Really, the whole process of trying to use AI with chemical design started off a long time ago. So chemical informatics, the study of kind of how we store chemical information in databases, was a big area of classical machine learning research back in the 80s, 90s, early 2000s. But around 2008, there was a very famous paper from a group in Canada who decided to what if we try to embed compounds, chemicals, kind of molecular graphs that you've all seen in chemistry class, into something that we can actually compute on with neural networks. And this paper sort of kicked off a whole massive amount of investment in this industry. And here we are today with kind of hundreds of startups all around AI and chemistry. And it's one of the primary test cases for a lot of the new HPC machines that come online. So just a little bit about my research really quickly before I give you just kind of some background on how I think about applying AI and HPC to drug design. Some of my work has focused on trying to speed up drug discovery workflows. So how do we use the full scale of machines to identify new chemical compounds? So this work was a big part of the COVID effort with the National Virtual Biotechnology Lab, where we found an inhibitor of the main protease. And my work also kind of addresses some of the other concerns, which are, well, a lot of times with HPC, we have these big monolithic code bases, but how do we actually interact with these, and how do we think about them in terms of how good they are, the economics of them, and even how do we interact with them visually? So the basic problem for drug discovery is that finding a new therapeutic costs billions of dollars, and it takes tens of years and a very long time. And given recent events with pandemics becoming a bigger concern and more of them occurring, this kind of time frame is not a great one to have. And so the idea is, how can we leverage AI and some speed ups from the AI industry to try to accelerate the time it takes to identify a disease, identify a target for a disease, and then actually move on to computational screening. So the way that I think about drug discovery is that it's a massive funnel. So in theory, there's like over 10 to the 67 drug-like molecules, which is, more than there are stars in the universe, kind of, all these kind of astronomically large numbers. And so it's generally thought of as like a funnel where we take this kind of massive design space of kind of chemical graphs, and how do we kind of narrow it down to some set of compounds that is tangible and realistic for us to test in a lab setting, say in cell lines or small plates, and then even the smaller set that we can actually bring to clinical trials, which are expensive and long running, and then all the way up through FDA approvals. So the basic idea of just kind of the 101 on how drug design works is disease is oftentimes caused by overactive or inactive proteins in the body. So one thing that we like to do is we could say, all right, well, we know that there's a particular protein that's cause of this disease. So for COVID, the main protease, for example, aids in the replication of the virus. So if we could find a molecule that fits in the pocket, in the binding site, in this little kind of flexible region of a protein that inhibits its action, then we can effectively stop the disease. So one of the first challenges is, how do you identify binding sites? So this is a whole body of research, and we have many different techniques for doing this, but let's just kind of assume that we have a target in mind and we kind of have a binding site. So there's a traditional tool in computational drug discovery called protein ligand docking or structural docking. And the basic idea is like using a lock and key. So you can imagine that binding site is kind of like a key or a lock, and you want to find a compound that kind of fits in there and stably sits in it. And so there's some classical techniques using traditional computing, like single processor CPU computing, that will take a molecule and kind of simulate it in 3D space and try to place it in different orientations to see how well it might fit. And so this was a bottleneck in traditional computing because it's CPU bound. There's only so many cores we could fit on a die, and there's only so many of these cores that we can fit in kind of one big machine and actually manage through something like MPI quite effectively. And so one question is, well, can we replace this with a surrogate model? So a surrogate model, if you haven't kind of talked about this yet, it's just an AI piece of code, a neural network that replaces traditional CPU-bound code. And so one thing that you might want to estimate using this surrogate model might be kind of how well a molecule might stay in place. So there's different techniques for measuring this. A common one is free energy of binding, which basically says, is it more likely that a compound will stay bound to a protein, or is it more likely than not? And so we might want to say, create a model that is able to predict this task. So you can see here's a simulation of a compound. You can see how the lock and key analogy kind of breaks down a little bit because, of course, everything's flexible. Everything's under different types of forces going on. And so if we have, say, a data set that we say we run a bunch of simulations and kind of bootstrap a model from, how can we fit this model into a big workflow? And so there's a couple of different ways that these workflows are theorized. Some of them might be you take a molecule, you maybe plug it into an ML surrogate instead of using a traditional one, and you can get to a goodness or a fitness score of this molecule much faster than using the traditional one. But let's look closely at the way that these workflows are conceived of. So one thing that you might think about is you have some type of property prediction. So this property could be how well it fits. It could be, is it toxic to the liver? It could be, is it easy to synthesize? And so one thing you might want to do is you just go to your favorite chemical vendor. They have tens of millions of compounds available, and you want to apply this model to it. So of course, this is a kind of traditional HPC problem of how do you scale a model across a bunch of machines? How do you collect all those inferences? And then how do you update this? So in this, we use what we call a layered workflow, where this top loop, for example, will take some candidates. So these could be molecules from a database, or they could be machine learning generated molecules, which I'll talk about in a second. And we might predict some property on them. We might score them, kind of keep them around for later. And then if we find some compounds that score very highly, we might say, well, let's pass these along to some experimentalists down in the other building here at Argonne, and let's have them actually apply it to some cells, apply it to some live virus or some functional assays, and see what we get. And so you can use those results to then update your property models to kind of shift from just using that bootstrapped simulation data to kind of using some real experimental data. And you can imagine how we can kind of run this loop over and over again and kind of say, hey, well, let's generate more compounds. Let's refine our models based on those experimental results of where maybe our models were a little bit off, and keep going. And so one thing that's exciting about this is that it connects well with some large machines, because what you are able to do is actually run these two workflows at different timescales. So you can always run kind of these property models, just screening billions of compounds all the time and kind of looping over and trying to identify ones that are interesting while we wait for those experimental results to actually come in. So the way that I've kind of represented this in the past is one way you might want to think about it is if you have one kind of GPU, which kind of has some inference rate, which is relatively fast, much faster than a CPU, well, then you want to have another model that is able to actually predict properties just as fast. So if I have a neural network that's able to generate compounds extraordinarily fast, I need to have also a consumer of this pipeline that's also able to get things equivalently fast. And you can actually do these things extraordinarily quickly with AI. So for example, in our tests, and this is from a few years ago even, so it's probably much quicker now, we were able to say generate compounds at a rate of say 10,000 per second using a generative algorithm, and we were able to screen those with an equivalently fast speed. The problem, of course, then in drug design, as well as we want to get things more and more accurate and go down the funnel, you have to eventually deal with very expensive simulations that might take days to run. You might have to deal with even slower experiments that take days to run. And so one challenge is to think through how you actually are going to handle the rate of generation of different things going on in your workflow. So this is just an example of like using an RNN to generate possible candidates for drugs. And you're actually able to sample these RNNs extraordinarily densely. So you'd think if you just have a small network and you train it on, say, here's 100,000 compounds, can you show me some more like it, that you wouldn't be able to sample it that densely. But we found that you were able to really sample this to like 10 billion compounds, 100 billion unique compounds, all with little to no redundancy. So during the COVID effort, we were actually able to use a workflow just like the one I was talking about, where we kind of prepared a bunch of proteins based on COVID targets that we were developing at the time using homolog models. This was in early 2020, before the structural biologists were even able to get the real structures. We were able to screen these on the kind of the 10 million compounds that were available to be purchased within a few weeks. We were able to use AI to do this extraordinarily fast. And within a month or two, we had compounds already in the laboratory being tested against a virus. And we actually ended up finding some, which was kind of super exciting. These compounds are still kind of in the whole development pipeline of drug design and being optimized, which is kind of non-computational at this point. But of course, I'm doing some work to try to bring it into the computational way to run a little bit faster. And of course, one thing also is that you can also scale this up for all different types of disease and make this workflow even more complicated. So you could start to add in things beyond just a layered property prediction, where you bring in questions about toxicity, you bring in questions about generative sequence design. So say if we want to think about how COVID might shift in variants, what might that structure look like if we look at the evolutionary history of it in order for us to design drugs that might be stable? Or even for cancer, you could think about how cancer can become quite resistant to single target drugs. So how can we think about doing even targets where we design, say, a drug that might fit two different ones and have a kind of game theoretic approach to trying to outsmart the kind of evolutionary tendency of drug resistance? And of course, I think it's also important too to point out that equally important to developing large workflows is also developing ways to interpret those large workflows, interact with them. And so there's a PhD student at Northern Illinois University who's been working on developing an interface that actually allows us to, in real time, kind of analyze those properties in generative models while a workflow is going on and be able to interact with them in a kind of visualization. So that was kind of a quick science talk on AI for drug discovery, and I hope I might have given you some new ideas on how you could take some science applications, theorize them a little bit with workflows, and then apply it to your work.
Thank you, Austin. So what we'll do now is we have a little bit of time for questions. I think I see one potentially coming in Slack. But while that sort of while folks potentially populate Slack with questions, I have one question for you. So obviously for something, actually one just popped in, so we'll go for this. Catherine asks, do you check to see if a compound that fits well into the protease also fits into other proteins that are in the body for side effect purposes?
Yeah, that's an excellent question. And that is something that we've thought about doing and we've still yet to implement. There's a few reasons for this. One is that there's tons of proteins. So being able to screen, even 1,000, 2,000 different proteins would require us being able to identify where on those proteins we think the drug will bind. And so there's been some advances in trying to detect different in an automated way, given just a protein structure, how can you find a binding site? But we haven't had a chance really to look into that yet. And especially now with AlphaFold, I think there's a great opportunity to include things like off-target effects in the research pipeline up front.
Thank you. So another question from me. So obviously for something like COVID coming in 2020, I have to imagine that the data you had to train things was probably more limited than it probably is now, right? So when you're going into solving problems, especially that are like relevant right then and there, where's your data coming from, and sort of what are you using to sort of train the systems that you have in ways that are also accurate and hopefully will save people and lives?
Yeah, that's a wonderful question. And so I think that in computer science, we oftentimes have a kind of luxury in some ways that might be different than the sciences, especially in kind of computational domains where we can run simulations. Oftentimes we can generate very large data sets from kind of bootstrap data. But of course that data is from simulation. And so then you're kind of training a model that's slightly, off distribution, you could say, from what the experimental data is. And so during COVID, this is what we did. We didn't even have structures yet, but we kind of knew from previous versions of SARS-CoV what they kind of look like. And so we started all of these kind of ML pipelines based on we think it kind of is going to look like this. And this is kind of what the results we get from these simulations on that kind of accurate data. And then over time, as we got more data in, and this is the kind of active learning part, we were able to fine tune these to say, well, given this data that's a little bit different than one that you've been trained on, can we shift the distribution over a little bit?
Sure. You also talked a little bit about sort of connecting to the experimentalists, which I imagine can be hard. Are there challenges that you have come across when you're like actually trying to then translate this, something that you find computationally, to the benchtop itself?
Yeah, of course. One big challenge is sometimes, and this one kind of goes unspoken, I think, a little bit, is that you actually have to convince experimentalists who want to work on those projects and want to do those experiments. Experimentalists work on thousands of wonderful science projects all the time, and here comes a computer scientist who says, I have this computer thing that says that we should do X. And so it requires a little bit of working back and forth to both convince them that, there's some trust in the system, that, there's trust in us too, that, we're not just kind of turning a crank and sending you things to, waste some amount of your time and money on. So that's one challenge. And I think the second challenge too is that there's oftentimes in experimental science a bunch of caveats for doing experiments that are hard to articulate and codify. One example is in some work that we're doing with creating peptides for looking at bacteria that are resistant to standard antimicrobial therapies. Is we automatically generated a bunch of these peptides and simulated them. And so then we go and kind of do the classic experiment where we just try to synthesize them, put them in a plate with some bacteria, and measure the change in growth. But one thing that we did not account for is that not all peptides are soluble, and sometimes they aggregate. And so when we got this data back, we saw that half of the plates were just kind of clumps of white protein matter. And this completely threw off the data. And if we would have kind of worked more closely and thought about that we actually had to do this physically in the real world, then we should have incorporated more controls about, say, solubility, about aggregation, and ahead of time. So sometimes starting the conversation earlier can help with some somewhat embarrassing experimental results.
Sure. Another question, this one is from Jianan. Jianan asks, is there a trend for using some sort of specific descriptor for representing the molecular proteins for machine learning, or something like a graph neural network?
Yeah, so there's all different types of approaches to the way this is done. When I started working on this in 2008, we primarily were using descriptors at the time. Then as kind of language modeling and sequence modeling grew in like 2019, 2020, we started using SMILE strings, which are sort of just searches on a graph that kind of show the graph structure of a compound as a sequence. And nowadays, graph neural networks are quite common where you just use the graph structure directly. But so far, there hasn't been, I think, kind of a community opinion on what's better or like what's not. I mean, it's a really hard question to ask of how do you prove one representation is superior than the other, especially when the methods that you use on those representations vary.
Yeah. Another question from Thomas. How much do you find you have to tune the models and pipelines to the specific problem that you're solving? And how much are you able to use those things just out of the box?
Yeah, that's a great question. So in theory, these things should be a lot more applicable generally. But oftentimes what happens in practice is a lot of these experimental and kind of protein-specific considerations get kind of creeped into the code in some way, which ultimately kind of hinders the ability to just use it out of the box again. And so I think kind of working on better software practices in some way is one way around this, of kind of being very careful about how you think about what is problem specific versus what is more generalizable, and making sure the way that you write your workflow and code kind of modularizes all these pieces. So like in 2020, when we were trying to do this as fast as possible, this was not at all what happened. And so now we spend a lot of time trying to go back and generalize this for other projects we work on, like in cancer or for bacteria.
Sure. Okay. So one final question. This one is from Catherine again. Are there ways you can build your system so that new data from experimentalists can be incorporated more easily back into the machine learning models?
Yeah, so one challenge is experimental data, as I said, is extraordinarily messy, and a lot of times it requires interpretation to really understand what it means. But there are some things that I think experimentalists can do to also help out with this. So one thing is that when they think about their protocols, sometimes they like to get very specific about a particular target or a particular way of doing an experiment. But if you're trying to say aggregate data, say you have some data from online from some previous study someone's done, and then you want someone to do some new data, trying to get someone to agree on a common protocol is I think, a big challenge, but it does extraordinarily help out, say, the modeling effort when you're not spending weeks trying to normalize different batches, which is something that, again, I don't think it's taught too much, but you actually spend most of your time doing.
Sure. Awesome. Well, I think we're out of time. Thank you so much for coming and sharing the work that you're doing and answering all of these questions. I think if folks have other questions for Austin, I'm happy to shoot them his way and sort of fill those out for you. But for now, I think we'll say goodbye to you and jump back over to Bethany.
Hello, thank you. Okay, I'm going to share my screen again. Okay, so we talked about you don't want to underfit, which is your model is too simple for your data, and you don't want to overfit, which is your model is too complicated for your data and it won't generalize to other data. And so one way to look out for this is that you have some test data and you hold it out. You don't use it for any of your training process. You hold it out till the very end. And it's common to actually split your data into 3 sets: training, validation, and test. So for example, it could be 70%, 20%, 10%. This could depend on how much data you have. You use the training data for that gradient descent process of fitting the parameters. The validation data, it can be used to compare different options, like different learning rates, different numbers of layers, different widths of layers, or deciding when to stop training. In the test data, you leave aside, maybe it's in a vault, and you do not check it until the end of your project. When you think, I have a good network, I think it's good. It has good training error, it has good validation error, and I want one final estimation of how well it'll generalize, and then you check the test error. And so in the context of research, this could be that you are ready to publish a paper. Now you check the test error. In the context of working at a company, this could be like you're ready to deploy the model for the first time. If you look at the test error repeatedly, then you're not really doing a true estimate of how well you can generalize. If you check your test error and you're surprised that it's way worse, then you could try to improve your model, but now you need new test data if you want to really estimate how well you can generalize to unseen data. And interestingly, so in the machine learning community, there's a lot of like common data sets, such as MNIST. And you saw that we imported MNIST and it was already split into a training and test set. And so like tons of people all over the world have imported MNIST with the same training and test split and have been using it for decades. So there's actually concern that in these like very common data sets, the whole machine learning community is overfitting that test set because it's the same test set that everyone's using. So maybe people think I have the best MNIST network ever, state of the art, and it's because of some kind of quirk in the test set. But the more common problem is just to make sure that you're leaving your test data till the end. Okay, so we're gonna do another in-class exercise. This time we're gonna go into breakout rooms so you can work with a few other people. Now, so far, we've just been looking at training error. The test data is around. We haven't touched it because that would be bad. But now I've introduced the idea of validation error. So the in-class exercise is to split that training data into two pieces, a training, what you're going to call training, and what you're going to call validation. And then change our training loop so that it's only fitting on the new training set. And you can also track the validation loss during the training loop. So we can plot both training and validation loss here. You could plot both training and validation accuracy, and we can see if there's a gap. If there's a gap, then maybe we're overfitting the training data. Okay. So Paige, you can do some breakout rooms.
Yeah, sure.
And people can add into the notebook here.
Yeah, so we're gonna take 10 minutes for these breakout rooms, and each breakout room will probably have 4 or 5 people. So please talk to your co-participants and see if you can sort of work through things together. So I'm going to open the rooms, and then I'll also ping you once we get started.
Thank you.
All right, welcome back, everyone. Hopefully you had a little time to work through things, and we'll sort of continue on. Bethany?
Oh, thanks. Okay. I think we should have given you more time because I have an error. We'll see if you guys have. Okay. So I gave you guys the hint that there's a train test split function in scikit-learn. They call it train test split, but of course we can call them train and val. So I gave them our X train and Y train. I stuck with their default suggestion of 1/3 be split off for test, which we're calling val, and their random state of 42. And just so I don't mess up, I called X train new and Y train new. Jupyter notebooks, we can create a mess of like losing track of the order of running cells. Okay, so now I want to track the loss and accuracy for both train and val. So I made more arrays to track them. We're still doing this sort of lazy grab one batch. In the loop, we do the training step. Then I get the training loss. Now I want the validation loss. As you can see, I have an error because I think I'm mixing that we kept changing the model. Now I'm at the bottom of the notebook, and so I have an error about size. I think we're outputting the model that outputs a 10-dimensional vector. And Y is just 1-dimensional, but I don't know why I got the error here and not here. But okay. Train accuracy, validation accuracy. This is what I was going for. I have an error because of my models being mixed up. Let's see if we can fix that by rerunning just the top cells to define the model.
Sorry, is there any reason why you would add a new access to the Y train batch definition at the top of the loop?
Sorry, let me scroll back down and see what you're referring to. Oh, this part?
Yeah.
We needed that for, I think, so sometimes NumPy you have the differences between your vector being, let's say like 20 comma, so just a one-dimensional vector versus shape 20 comma 1, a two-dimensional vector where the second dimension is 1. And for some reason I needed it to be 2 dimensions where the second dimension is 1. Up here somewhere. Sorry, I don't remember at what point. Maybe when it might have been when I do this, because I want the dimensions to match. Okay, does anyone spot my error? Confused about why the training works and not the validation.
We want
Oh,
to change X val and Y val to put into those new variables that you created on 37 in the cell above.
Yeah, your Y val doesn't have the new access. The broadcasting isn't working properly.
Of course, thank you. So this should not be taking so long. I don't know if I've done something crazy here. Okay. If no one has any suggestions of why this is taking longer than normal, I think we get the idea. Does anyone have any questions about tracking, training, and validation error?
So does this, because the way that this is split, it kind of splits the original training data into a new training and a validation. But this doesn't really include the testing split that that was mentioned above, right?
Right. So really, I would do this at the beginning of a project, and MNIST gave us, when we loaded the MNIST data, this helper function for loading MNIST data that this library already gave us a train test split. So it's like everyone in the world has the same test data for MNIST. And now we're like, well, it'd be nice to have a validation data set as well, so that we can use it to compare models, like compare learners. So that's why we're further splitting train into train and validation. And really, we would do this before we start training. So we would I would import MNIST and then immediately resplit the training data into train and val. And that way, when we fit the model, we're fitting on some percentage, such as 70% of the data. We can use the validation to see how things are going. So things like we can decide that we can stop the training because the validation isn't improving anymore, or we might be comparing hyperparameters like learning rates. Does that make sense?
Mhm.
Someone in the Zoom chat figured out why it was taking so long. Looks like you weren't batching the validation set. Saying it all at once.
Thank you. Yes. This is much faster before because we were doing just this small amount of the training data, but I'm using all of the validation data, so it's taking longer. Okay. Oh, Google Colab is giving us a warning that we're not really using the GPU. Because we haven't done anything to tell it to use the GPU. Okay.
This is kind of how you would split it. Sorry, what I posted in the chat would be how you would split it into like batches instead of using your entire training data set, right? Because now you're kind of splitting it the same way that you do for the training new batch.
Mm-hmm. So when we move to TensorFlow, you can set a batch size for how much of the training data to use at a time for each step. And there's also a batch size for validation data if it's too big to check all of it. But more commonly, well, I don't know what's more common. Another option is if the validation data is reasonably small and you're fine with checking the whole thing. You just check it less often. So there's no reason to check it every iteration. Okay, so next week we can talk about more complicated networks, and it'll be easier to use it because of Python packages. Now there's various universal approximation theorems, and roughly glossing over the exact math, they say that a nonlinear neural network with just one hidden layer would be like my blue ones here. So this is sufficient. They're for any reasonably smooth data, where here you have like formal math terms. There exists a neural network with one hidden layer that can fit that smooth function arbitrarily well. So it technically only one hidden layer is needed, but it might have to be very wide. So I know people were asking about width versus depth. So some trade-off of when you want to make your network more complicated, you can make it more complicated by making those layers wider or deeper. So the theorem says the network exists. Now, that doesn't mean it's easy to find with optimization. So one thing we'll learn about next week is convolutional neural networks. So they have layers that are more well-suited for images than just what we call fully connected, which is that we're just multiplying by a matrix. So that has that diagram of all those nodes are connected. So it's technically possible to fit an accurate network to this image data with fully connected neural networks, but it'll be more efficient with convolutional neural networks, which we'll talk about next week. Now, here with our training loop, we only had the linear network. Once we add all those non-linearities and introduce different loss functions, the gradients get more complicated. But I do have for you guys notebook number 2. And here I have a full implementation of a 2-layer network for MNIST. And I tucked it away in these Python files that are in the repo. So we're going to import from these files. You can dig around if you want to see the implementation of those gradients, but it's like a lot of calculus. And I think I ended up following the formatting of this homework from Stanford's class. They have you implement this 2-layer network, and they kind of broke it into pieces. It's like reasonably easy to follow along if you're familiar with calculus. And I remembered I forgot to change some of the comments in that code, so I'll fix that. So in this notebook, we import MNIST again. These are all these same pre-processing lines. But now you can use this nonlinear neural network. It has 2 layers, one hidden layer. And here you can decide how wide to make that hidden layer. And here I have just like a very simple implementation of stochastic gradient descent. There's more weights and biases to update with the gradient descent, so there's a loop here. And this would be one step of stochastic gradient descent. Here's our accuracy metric again. And so here's an example training loop. I'm not sure if we talked about the terminology of epochs, but in machine learning training, in each epoch, we're looping over all of the training examples. So you can choose how many epochs to go. Here I started with 10. Within each epoch, we're looping over all the batches. This is like a way to loop over all the batches. You'll learn later better ways to do this with TensorFlow. Here's one step of stochastic gradient descent with that batch. Check the loss. Here I'm checking the accuracy just once per epoch, and I'm printing to the screen at the end of this epoch what the loss and the accuracy are. And then I'm reshuffling the data at each epoch so that in the next epoch, when you go through the batches, you get a little bit more randomization. The examples aren't batched together in the same way. So some homework is to experiment around with this and see if you can improve the accuracy. Some easier things to try would be changing the numbers that you see right here in the notebook, like the learning rate, or more epochs and so forth. A more advanced thing to try is if you go into those Python files and add more layers. If you're feeling more ambitious, you can also experiment with like the width of R1 layer. This scale factor is when we randomly initialize those weight matrices. In this implementation, it's a normal distribution, mean 0, and some standard deviation. And so here I have 0.01. You could change that. The initialization of weights can be important. You can think of it as when we had this picture of a surface and you're doing gradient descent down, you can think of it as where are you starting on that surface. And so people have different heuristics for how you should, like what random distribution you should use. In this case, the implementation is a normal distribution, but you can change what the standard deviation is. Any questions about this? We have.
Yes, may I ask you a question?
Mm-hmm.
So you mentioned the criteria, how you define the overfitting and underfitting.
Mm-hmm.
So how do you when you say that the model overfits or underfits? What's like quantitative measure for that?
Oh, yes. Okay, so underfitting would be, so let's say you're working on this and you think the accuracy is not very satisfactory on the training data, like it's 50% accurate or something. You're like, oh, I'm sure you could do better. So that would be underfitting. The model isn't complicated enough to fit the training data well, or it's complicated enough that the optimization isn't going well or something like that. Overfitting would be there's a big gap between the training and the validation loss. So maybe you're happy with the training loss, but the validation loss is much worse.
Mm-hmm.
Thanks for asking that. It's a good question.
Hi, I have a quick question.
Mm-hmm.
So what is the highest accuracy you get on this data set?
Let's see. Anyone else can correct me if I'm wrong, but I believe with MNIST, it's quite possible to get 99% accurate, but I don't expect you to be able to get 99% accurate with this code.
4
the
nines,
model's too simple.
I think. Like 99 point, well, I guess it's like 1 or 2 misclassifications.
And that's on the test data.
Yeah.
Okay, thank you. Wow.
I think if you run the notebook the way it is right now, it's like 40% accuracy on the training data. It's quite bad. And I do not know the highest possible with this code, but it's definitely a much simpler network than you could have. So don't stress about getting 99% accuracy, just explore how to improve it. Any other questions in the last couple of minutes? Oh, so to submit the homework, you can submit your version of this notebook. If you're doing it in Google Colab, there's a little extra step so that Google Colab can load up those Python files. So I'll put that in Slack. Because when we do that thing where we search for this notebook in Google Colab, by default, it doesn't bring up all the other files in the repo. So these helper files I added. So I'll put in Slack the extra lines to add. But of course, if you do this somewhere else, like on your own computer or on ALCF, then it's going to know how to import the Python file in the same folder.
Okay, so I have to make sure that our notebooks basically run on another computer. So I thought the idea of submitting the notebooks was that we basically submit the results so that you wouldn't have to.
Yeah, I just meant for your sake. If you want to run this on Google Colab to do your homework, Google Colab won't by default import the whole folder from GitHub. But if you do this on your own computer, it will have the whole folder because you cloned the whole repo. Does that make sense?
Yeah, and follow-up question. Will there be so the homework for this week is just work on the full net notebook and try to get as high of an accuracy as possible? Will there be just to check, will there be a more formally written homework question kind of like we had last week, or that's just the goal for the week?
That's the goal. I can, if you want, I can put it explicitly here, but I put, yeah, I just put, can you do better, but I call it the homework. but I can put it explicitly there for people who aren't present today and are looking later.
That might avoid
Excellent.
some questions on Slack, yeah.
Thank you, Thomas. All right, well, it is now the top of the hour. So thank you all for coming today. Please, if you are looking to get the certificate of completion, please submit your homework using the Google form that is posted in Slack. And we will see you all next week. The recording for this session should be up in the next few days as well. So thank you all. Goodbye.
Thank you.